So thank you everyone for joining the CDTRP training webinar today. My name is David Hartel. I'm the Executive Director of the program. I'm based in Ottawa. And we host these bi-monthly training webinars really to give people something new in donation and transplantation research, to try and show them a different perspective or a different way of doing research or thinking about how we could develop our understanding of donation, solid organ transplant, or hematopoietic cell transplant. And so I'm very excited today about today's topic. I think this is something that's totally out of the box and a new sort of interesting way of thinking about transplantation and what this means, and taking some time to really test all of our brain cells and think about transplantation from a very metaphysical perspective. And I think that that should be very interesting. So I'm going to turn it over. I'm going to turn it over to Lauren Westhaver, who's going to do the introduction today. She's a MSc candidate in the Department of Pathology at Dalhousie University. And Lauren is co-supervised by Dr. Ian Alwyn and Jeannette Boudreau. And her research is focused on the role of natural killer cells and mitochondrial damage associated medical pattern molecules in the pathogenesis and regulation of ischemia reperfusion injury following liver transplantation. And Lauren is one of our CDTRP trainees. So Lauren?
Hello. I'm actually, I've recently switched over to the PhD program. So
oh, amazing. Congratulations.
Bio. Yeah. Okay, so today we'll be hearing from Dr. Ramesh Prasad. The title of his talk is The Philosophy of Transplantation. So a bit of background on Dr. Prasad. He obtained his MBBS medical degree from Osmania University, Hyderabad, India. He completed his internal medicine residency at Wayne State University, Detroit Medical Center, nephrology fellowship at the University of Pittsburgh Medical Center, and transplantation nephrology fellowship at Toronto General Hospital. He holds an MSc degree from the Institute of Medical Science, University of Toronto, plus MA and PhD degrees in philosophy from the University of Waterloo. He is a staff nephrologist and director of the kidney transplant program at St. Michael's Hospital, and a fellow of the American Society of Transplantation. Dr. Prasad's research interests include all aspects pertinent to the long-term health of kidney transplant recipients and donors. He is particularly interested in transplantation-related cardiovascular disease, the eradication of organ trafficking and transplant tourism, and the philosophy of medicine, especially the epistemology and metaphysics of organ transplantation. So clearly quite an impressive resume. And so with that, Dr. Prasad, you may begin.
Thanks, Lauren, and thanks, David. So my topic today is broadly the philosophy of transplantation, but what I will be doing is focusing on one particular aspect of transplantation that I think will be very helpful to solving two of the major problems faced by transplantation medicine, namely transplant tolerance and xenotransplantation. And though I won't be going into the scientific aspects of those problems, I'm hoping that in the end, through a philosophical perspective, and more specifically a metaphysical perspective, we can think of new ways to approach those two rather urgent issues in transplantation. So just a bit in terms of background, what is the philosophy of medicine? Because the philosophy of transplantation would be a part of the philosophy of medicine. And the philosophy of medicine is a huge movement right now. There is a philosophy of medicine society called the Philosophy of Medicine Roundtable. If you're interested in learning more about that organization, the Philosophy of Medicine Roundtable, you could, a simple internet search term will take you there. So there are three broad areas in philosophy. So there is ethics, there is metaphysics, and then there is epistemology. And as medical scientists and medical practitioners, we're most familiar with the ethics part of philosophy. So that's the typical extent of our interaction with philosophy. We've all interacted with bioethicists, and we have bioethical problems to deal with in medicine, perhaps even on a daily basis. But philosophy of medicine tries to distinguish itself from bioethics. So instead, the focus is on the metaphysics and epistemology of medicine. And so just a quick reminder, metaphysics deals with the nature of reality. So what is real, what is unreal, and what goes into justifying what you think is real and what is unreal in this world. Whether the world itself is real or not is a question that comes under the purview of metaphysics. Epistemology relates to the nature of knowledge. So what is knowledge? Is it justified true belief, as Plato would have us believe? Is it justified belief? Is it true belief? Is it some combination of them? How do you know that you know something without going into a circular type of argument? So that comes under epistemology. And today's topic comes under the purview of metaphysics, the nature of reality. And I will be delving into what the human being is really all about. So what are some of the common topics given at the bottom of this slide here that the philosophy of medicine addresses? So what is health? That's actually a very difficult question. And if you ask 10 different medical practitioners, and if you ask 10 different philosophers, you're going to get 20 different answers for what is health. Similarly, what is disease? What is a syndrome? What is a cure? What is disability? What is discovery in medicine? What does it mean when you say that you have discovered something? What is an explanation? What counts as an explanation of health or disease or a process? How do you measure health? How do you measure disease? What is clinical judgment? What does it mean when a consensus guideline comes out about some particular disease process or the other? What is the role of evolution in health? What is the role of evolution in disease? What is the role of evolution in the structure of the human being? And how does that affect how medicine is practiced, and so on? So these are some of the topics. These are all not ethics topics. These are all philosophy of medicine topics. So here's an example of a solid organ transplant, a kidney transplant in this case. I'll be sticking to transplanted kidneys primarily, only because the kidney transplant is a technically simple procedure, and a lot of the basic principles related to philosophy that kidney transplantation can be used as an exemplar for also apply to the other organs and things like immune processes and so on. So we'll be sticking to the transplanted kidney here. But what I'd just like you to take away from this image is the notion of a boundary. So where does the transplanted organ begin? Where does it end? And where does the host begin? And where does it end? Because that's going to be crucial to our understanding of what transplantation means in a metaphysical sense. So what was my motivation for the project that I will be discussing today? Much of medical knowledge relates to the basic biological processes involved in supporting the healthy functioning of the human being. That's really what we strive for with medical knowledge and why we spend so much time and energy in the pursuit of medical knowledge. We also know that organ failure causes death. We also know that organ transplantation treats organ failure, and it's one of the most profound medical developments in the last century. The immune system is essential to health. The immune system, however, opposes transplant success, and this is a quandary we face as transplantation physicians and surgeons and scientists. We have this immune system that is essential to health, but this very same immune system opposes the success of our transplantation, which is a therapy for improving patient health. And so we need to understand why the immune system opposes a life-saving therapy. And if we can understand why the immune system, which we need for health, opposes a life-saving therapy, we might help to improve transplant success, because we view the immune system as something that interferes with what we're trying to do. But in order to do so, we need to understand the basic biological processes of health. And so in order to do so, solving some of transplantation's problems, things like xenotransplantation, transplant tolerance, requires looking outside of science and medicine. And what I'm looking for is a solution to these sorts of problems through the lens of philosophy.
So Ramesh, sorry, just going to stop you. There seems to be something right we keep getting a warning message saying that there's a window and you need to move a window away from the shared application.
Okay, so
Not
any
sure what
idea
that is.
how, yeah. So you're seeing this yellow box in the center of
Yeah.
the screen?
Yeah.
Let me see what we can do.
Sorry.
Ramesh, are you running 2 monitors?
Am I running 2 monitors?
Yeah, on your computer.
Okay, so hopefully I won't exit.
No, that's okay. Sorry, I didn't mean to interrupt the flow of your presentation. It's off to a
No,
great start. I just wanted to make
okay.
sure that.
Okay, I'm going to take a risk here and close one window. So let's see what happens.
Okay. Aha, success.
Worked. Okay. Perfect.
Hold on.
Can everyone see the slide?
No, now I just see a green screen. There we go. Okay, we're back on track.
Excellent. So what I will be doing is moving from the bottom to the top, starting with our understanding through a philosophical perspective of what a species is and what an organism is, and then moving on to the concept of a superorganism, how immune responses tie into superorganisms. And then we'll talk about emergence. Emergence is an important concept in philosophy, and that certainly applies to transplantation. And then we'll see how this all ties into health and disease. So let's talk about species and organism first. So when we think of organism, we tend to use individual organisms as paradigmatic examples of individuals. So in our common language, we talk about an individual lion and equate that with the lion as an organism. We talk about a herd of giraffe, with each member of that herd being a distinct organism. But it gets a little more complicated when you think of species like ants. When you have a colony of ants, the ants aren't necessarily making decisions on their own. They're making decisions as part of a collective. And if you disrupt the relationship of the members among that collective, the members tend to move in unison based on the message that they are receiving. This also applies to termites. Termites are greatly dependent on the mound of mud and earth that they construct. They construct what are called fungal gardens and interact in a symbiotic manner with these fungi. And so the question there is, which is the individual and which is the organism, becomes philosophically quite interesting. And then we talk about individual human beings, and we tend to think of ourselves as individual human organisms as well. But as I'll show you in the next few slides, that is not quite the case. So is a human being an organism? Is a human being an individual? Or is a human being both an organism and an individual? So what exactly is an organism? So an organism has to have some kind of a boundary. There has to be some point beyond which the organism is not there, and there has to be a point in space within which all of the organism is there. And that distinction happens at the boundary. There is a physical chemical nature to the organism. It is constructed out of matter. The organism either is living currently or had to be living at some point in time. So an organism placed in a jar of formaldehyde in the laboratory would still count as an organism because it did have life at some point, and it also had the potential for reproduction, which is another important part of the organism's characteristics. Quite pertinently, an organism uses energy, and it uses this energy in order to resist decay and to persist in space and to persist through time. That is one of the key features of an organism. And it does so by exchanging resources with the environment. So there's always a back and forth. Stuff is always going into the organism, stuff is always going out of the organism. And if that stops, then the organism decays and ceases to exist. Now, some views of organism also count lineage. In other words, you come from your mother comes from her mother comes from her mother, and so on. And every organism would have a lineage in that respect. But there are organisms that resist that kind of a definition. So, for example, viruses are constantly mutating, and they mutate to the extent that they become unrecognizable from their ancestors. Bacteria undergo what's called lateral gene transfer. And so genetic material is constantly exchanged among bacteria. And so in a genetic sense, bacteria lose their identity when they do that. But at the same time, they persist in time and they are capable of reproduction. Autonomy again becomes an issue. Are organisms capable of living, persisting in space and time without the presence of other organisms, either of their own kind or not? And there are exceptions to that. So this is broadly speaking what an organism is, and the entire textbooks in philosophy have been written about this. And so species, in addition to organisms, essentially includes organisms with a common characteristic. They are capable of getting together and reproducing and producing more of the same organism as a result. So where do superorganisms come in? So I'm going to give you two examples here. The first one is the slime mold, or Dictyostelium discoideum. Now, Dictyostelium discoideum is an amoeba. It's an amoeba that lives in the soil. And for the greater part of its life, shown in the top left-hand column, the amoeba is happily engulfing bacteria and doing its own thing. But in times of scarcity, what happens is the amoeba coalesce. They coalesce and form this huge structure that you see at the bottom, visible to the naked eye. And this structure, called the slime mold, is capable of migrating across the soil, across significant distances, to areas where there is a more favorable environment. And at that point, it produces these spores. These spores then disperse with the help of the environment and then form new amoeba. So the question is, what counts as the organism? Is it the single amoeboid cell that's in the soil? Or does this count as an organism? Because this would seem to meet all the criteria for the organism as well. Are there two different kinds of organisms, or what exactly is going on here? It serves as a counterexample to what we know of organisms. Another example is the Hawaiian Bobtail Squid. So the Hawaiian Bobtail Squid, otherwise known as Euprymna scolopes, lives in seawater. And it has a particular characteristic of bioluminescence. Now, this bioluminescence makes it virtually transparent and invisible to predators from above. And it also makes it relatively less visible to its prey below. But this bioluminescence occurs as the result of an interaction with seawater bacteria, Vibrio fischeri. This is an organism that lives in seawater, and what the bacteria do is they migrate into the squid, into a specialized organ, and it is the interaction between Vibrio fischeri and the light organ of Euprymna scolopes which gives it its bioluminescence. And obviously this is beneficial to the squid only during the daytime when there is sunlight coming in from above. It would be harmful at night because you wouldn't want it to be visible to prey. And so every night the Vibrio fischeri leave the bobtail squid and the organism becomes dark again. And so the question philosophically here becomes, what counts as the organism? Is the bobtail squid one organism? And are the Vibrio fischeri all together one organism? Does each Vibrio fischeri bacterium count as one organism? Or does the whole combination count as one organism? So again, if you try to apply the definitions for organism versus individual, you'll come up with different answers. And bringing it back home to humans, we have microbiota. We have microbiota in the nose, in the mouth, the skin, the reproductive organs, the small and the large intestine. We even have bacteria in our stomach and of course in our throat. And so when we think of the human organism, what we're really thinking of is tissue derived from human DNA, from some kind of a human ancestry, with the microbiota being other organisms. But the fact of the matter is the human being, the human organism cannot live without its microbiota. And if the definition of an organism is persistence through space and time, then one really can't exclude the microbiota from the definition of a human being. And so superorganisms, which is what the human being really is, it's a human superorganism. It consists of physiologically integrated organisms of the same or different species, and these species have different lineages. Superorganisms are localized in space and time, and this localization occurs through structural boundaries as well as functional boundaries, and we'll talk a little bit about that in the next slide. But how are these boundaries maintained? And these boundaries are established and they are maintained by the immune system. There's communication, constant communication back and forth amongst the various organisms that constitute the superorganism. But because they aren't really of the same, they're not the same organisms, they don't fully integrate with each other. And so there is an inherent disunity of cooperation and conflict. So just to bring that back very briefly to the human being, when the human being dies, the microbiota does not die at the same time. And so the microbiota quickly gets to work decomposing the human organism. This decomposition starts in the intestinal area where there is the largest bacterial load. So the intestines are the first to rot in, sorry if anyone's eating their lunch. The human being is the first to rot in the intestinal area, and then that spreads through the whole body because each constituent organism of the human organism is pursuing its own life path. And while it's living in a relationship with the human organism, once the immune system fails as a result of death, the human organism becomes food for the microorganisms that are associated with that human organism. So there is a boundary. There is a boundary between the human organism and the microorganisms. And so what exactly is a boundary? A boundary is a very interesting problem in metaphysics. And thought about what a boundary is goes back all the way to Aristotle. And so his definition of a boundary is there's a limit. And so we call a limit the last point of each thing, that is the first point beyond which it is not possible to find any part of the thing, as well as the first point within which every part of the thing is. So as long as you're within the boundary, everything that is there inside the boundary belongs to whatever that thing is. But the moment you cross that boundary, nothing really belongs to whatever that thing is anymore. And so we have two types of boundaries that are structural and functional boundaries. Structural boundaries delineate the organism. So the human organism has its own boundaries. Functional boundaries, however, delineate the superorganism. So it's important to conceptualize that the gastrointestinal tract from the lips all the way to the anal sphincter, it's one continuous lumen, but that is not part of the human organism. It is part of the human superorganism because that is the lumen within which all the microbes are contained, but it is not part of the human organism. And so the bacteria and various other microorganisms that are present within the lumen of the GI tract are in some kind of a functional relationship with the human organism. Obviously, they cannot get too close or structurally invade the human organism because otherwise they would cause disease. But at the same time, they have to be near enough to the human organism to be of some benefit to the human organism. So structural boundaries separate organisms. Functional boundaries physiologically integrate superorganisms. There's a physical boundary in the physical barrier in the case of structural boundaries, no such thing with functional boundaries. Structural boundaries serve to inhibit communication, which is why you need pumps and channels and gates and all kinds of things to regulate movement across cells. Functional boundaries, on the other hand, either inhibit or promote communication. When structural boundaries are disrupted, there is an immune response. So the moment a microbe invades the intestinal wall, there is an immune response to that invasion. In the case of functional boundaries, however, disruption of the functional boundary does not always lead to an immune response. So billions and billions of microbes are leaving the gastrointestinal tract each day with each fecal movement, and billions of microorganisms are entering the human superorganism each day, and this goes virtually unnoticed. Functional boundaries develop first, and they develop from metabolic co-advantage. But on the other hand, if functional boundaries persist long enough through evolution, eventually they will develop into structural boundaries. And so now that we know what species are, what organisms are, what superorganisms are, and boundaries are, we'll now talk about immune responses, because immune responses tie into boundaries. And so this is what we're familiar with as transplant scientists. We have the antigen presenting cell, and we have the T lymphocyte. And when we talk about the immune response, in large part, we are talking about the various interactions between the antigen presenting cell and the T cell. And so how does the immune system handle microbes, and how does that compare to how the immune system handles transplants? So microbes have co-evolved with human organisms over millions and millions of years. So for example, the malarial parasite is human specific. And as human beings evolve through time, the malarial parasite also evolved through time, so that it depends on the human being for its reproduction and for its sustenance. And then you have other incidental microbes that can survive with or without the human being. And in jungle law, you can have plants and the insects that eat those plants co-evolve in ways such that the plant is producing toxins to deter them from being eaten. And various insects adapt to those toxins in ways that they could still continue to eat those plants. And eventually it gets to the point that you can have one insect that can eat only one kind of plant. And so that kind of co-evolution is occurring all the time in nature. With transplantation, however, there was no plan. There was no plan in evolution at all for transplants. And so we're completely redefining what's happening with the immune system. Microbes cross functional boundaries. So as I mentioned earlier, they're going in and out of the gastrointestinal tract, the respiratory tract, et cetera, skin, et cetera. Transplants cross structural boundaries. We're cutting through skin, we're cutting through subcutaneous tissue, muscle, we're entering the abdominal cavity, either intraperitoneally or retroperitoneally, and we're putting in an organ there. We're crossing structural boundaries, something that microbes, when they do so, only cause disease. Because when there are two possible outcomes when a microbe crosses a structural boundary. Either it continues to proliferate and continues to disrupt structural boundaries, in which case it causes disease, or its own structural boundaries get violated and it gets destroyed, in which case that process is called digestion. In the case of transplants, crossing structural boundaries leads to an immune response. The immune response in the case of transplants is adaptive. It's based on the adaptive immune system, while in the case of microbes, having evolved over a much longer period of time, that response has become innate. Antigens are presented by the recipient's cells in the case of microbes. In the case of transplants, these antigens can be presented through direct or indirect pathways by the donor or the recipient antigen presenting cells. With microbes, boundaries are repaired. So the disease is curtailed, the invading microbes are destroyed, and the boundaries, by being repaired, the original organism returns to its to the state it was before the invasion took place. With transplants, boundaries are certainly repaired, but because this transplant is persisting through time, these boundaries are redefined as well. And so that is the source of many of the issues that we face with transplantation. So you have microbes, you have organ transplants, and you have human organism persisting in time. And all of these are happening at the same time. We shouldn't lose sight of the fact that the human being, the human organism that is receiving the transplant, also has microbes. And so how do we understand rejection in this context? There is no willing acceptance of an organ by the human organism, just as with microbes. And while transplant rejection is an undesirable phenomenon, it is also a physiological phenomenon. It can be reproduced in vitro systems. And we know that if we excessively suppress rejection with powerful anti-rejection medication, this leads to other problems in the superorganism. Certain microbes may overproliferate and cause disease, or diseases in other organ systems of the human organism will occur. And so while rejection represents success in the self-preservation of the human organism and its environment, it really is a failure of the human superorganism to reintegrate. So we've violated boundaries. The human organism somehow has to deal with this transplant that is placed within its anatomical confines. It's doing its best to reject it. We're suppressing that rejection with medication. So in that sense, we're succeeding, but when rejection occurs, we're failing in that sense. And we're failing because we haven't taken into account the human superorganism. And so consider a superorganism where the largest organism is the human organism. And being anthropocentric, we consider the human superorganism as one single human being. But what it really is an integrated entity consisting of the human organism and all these smaller organisms. We'll consider these microorganisms. And a transplanted superorganism consists of an organ from another human placed within its structural confines with all the other organisms still present, outside the boundaries of the human organism, but still present. And so how does transplantation add to the study of superorganisms? The important thing is that the boundary between the organ and the organism maintains, because the organ, after all, is there functioning, doing its thing. We have the advantage of being able to study the immune system because there is a time zero with transplantation, something which we don't have with disease processes. We don't know when the first bacterium invaded the human organism in the case of a bacterial infection, but we do know when a transplant actually occurred. That gives us some predictability. In addition, there is some predictability to the immune response because the molecules of the organ, which after all comes from another human, and the recipient are more similar. We've developed pharmacological therapies to deal with this adaptation to allow for this to occur. And this occurs without mutation, which is what would happen in microorganisms to deal with new environments. With transplantation, we have the ability to study phenotypic responses in the target. We do biopsies of the transplanted organ, and we can study what happens. We can take the blood of the recipient, which isn't associated directly with the organ, and study that with different kinds of tests. We study urine biomarkers. We study all sorts of things to try to understand what is going on in the organ. And so what transplantation provides us is an accessible and coincidental structural and functional boundary between what counts as self and what counts as non-self. We don't have that benefit with microorganisms, where the structural boundary is different from the functional boundary. In the case of a transplanted organ, the structural and the functional boundary come together, and that boundary is what defines self and non-self. And so we'll talk next about emergent processes. So what is emergence? So I'd like you to think of some sand on a beach. So the sand on the beach has got millions and millions of grains of sand. But is there any emergence taking place there? So if emergence is to take place, think of another example of water. So water consists of two atoms of hydrogen and one atom of oxygen. So hydrogen has certain properties. Oxygen has certain properties, but water, which consists of hydrogen and oxygen, has the properties of neither hydrogen nor oxygen. It has its own set of properties. And so that is an emergent property, something that cannot be explained by the properties of its constituents. If you look at a beach with lots of sand on it, the system property would have to be invariant with intersubstitution of its parts with one another. So if you took out a scoop of sand off the beach, that beach is still pretty much the same. And even if you replaced it with another scoop of sand from another source, that beach really has changed very little. Also, size scaling of qualitative properties occurs even though values may differ when parts are substituted. So you might put a little bit more rocky sand in there, which is a bit heavier, but still the beach is essentially the same. There's invariance under decomposition and reaggregation of the parts. And there's no cooperation and no inhibitory interactions among the system. So the grains of sand are really not interacting with each other in any way. They're just there sitting next to each other. So whatever property you're thinking of, if one or more of these 4 properties is not fulfilled, then the property is said to be emergent. And the example I gave you is of water. So with transplantation, you now have a new part. This part was originally from one whole, one human organism. It's been taken out and it's being put into a new human organism as part of a new whole. And its function really depends upon its relationship to other parts of the organism. So when you think of a kidney, you really don't know if the kidney is working unless it is connected to a ureter. Without a ureter, you have no way of knowing if that kidney is actually working or not, whether it's producing urine. And that's true for interactions between the heart and kidney, the liver and kidney, and virtually every organ. And so in the case of the immune system, there's a continuous two-way immune interaction between the organ and the rest of the human organism. And this interaction occurs as a result of forcibly redefined boundaries. Emergent properties result from self-reorganization that occurs at the boundary. And we notice a number of emergent phenomena in multiple organ systems in a transplant recipient, some of which are unique, which you see only in transplanted individuals. And so therefore, the transplant recipient is a redefined superorganism. And so now you have the human organism that's received the transplant. Some changes have occurred in other organs in that transplant recipient. And then some changes have occurred in some of the microbes in the bowel and other places as well. And we see scientific papers published on a regular basis about what happens with the microbiota in transplantation. And changes occur in the organisms as well that we don't know about. We don't look for them. We don't measure them because they're not interesting to us. But changes are occurring because this superorganism has come to be redefined. And so think about it as blowing a bubble with soap. So what happens is when you blow air through physical force, boundaries are being created. We focus our attention on what's happening with the human organism after transplantation. But when boundaries are disrupted, as they are here with blowing air, other unexpected things happen and new boundaries are created. New, smaller, in this case, bubbles are created with their own sets of properties and their own boundaries. So you really can't predict what's going to happen when boundaries are disrupted, how they are going to repair, and what new entities are created as a result. And so that leads us into health and disease. So health and disease are both emergent processes. So you have a bunch of organs which are connected together in some way. And we use whatever criteria we can in a descriptive sense to describe that combination of organs as being a healthy organism or an unhealthy organism, as a healthy organism or a diseased organism. So these are emergent properties that really depend on the relationship of these organs among themselves. And these, in the case of transplant recipients, really occur as a result of redefined biological boundaries. And so health and disease can be better understood when we study the superorganism rather than the organism. And we do know that many diseases have a microbial or an immune basis. More and more diseases are being counted as related in some way or the other to the immune system. Of course, there are diseases that don't relate to the immune system, but many of them do, and that list is growing. And so the human organism superorganism distinction is a useful heuristic to study health and disease. And when we think about normative accounts of health, in other words, what health should be, we aren't usually considering actual events that are happening in the organism or in the superorganism. So there are a few quandaries in transplantation. One is that the organ transplant recipient is healthier than a patient with organ failure, obviously. But the fact of the matter is organ transplant recipients are actually healthier than many non-transplanted people as well. And even though transplant recipients are healthy, we are still, in some sense, in the back of our minds, considering them as diseased, even though the transplanted organ is healthy. And that transplanted organ, in the case of a kidney, may actually be functioning at a higher level than organs in people without transplants, but we still don't consider them to be healthier than those without a transplant. We also see emergent processes not seen in other human beings. So with kidney donation, for example, the kidney function of the remnant kidney expresses itself to a greater degree than it otherwise would have if donation had not taken place. And we know that transplantation does not cause functional impairment or a limitation. It actually promotes these. We look at transplantation as being a positive event because the starting point for change towards the transplanted state was not a healthy one. But in order to understand that better, we have to keep in mind that a disease process, whatever we're looking for in terms of diseases in a transplanted individual, there are actually multiple processes leading to a common phenotype. And so with transplantation, we're constantly balancing infection and rejection. We're going back and forth from health towards disease in one respect and towards disease in another respect. And so health is an emergent process arising from the mode of organization of the superorganism's parts. And so this is a cartoon of a macrophage, which is ingesting a tuberculin bacterium. You have a giant cell here, which can occur as a result of such ingestion. And then the example that I gave earlier on in this presentation about Dictyostelium discoideum turning into a slug. And so the immune system produces an outward immune response to microorganisms and an inward immune response to the transplanted organism. But what it really is a response within the superorganism in all directions. And so the health of the superorganism has to be considered differently from the health of the organism. But unfortunately, we are able to record only disease features in the organism. And so in transplantation, if we are to achieve tolerance, tolerance represents harmony between the organism and the superorganism. And that is something that we haven't been able to achieve yet. And unfortunately, in transplantation science, our therapies direct more to the organism, while really we have to be directing our therapies towards this superorganism. So to summarize, superorganisms are integrated organisms. Humans, when we talk about a human being is an ambiguous entity. We are really talking about a human superorganism if we are talking about the human organism in conjunction with its associated microorganisms, or we're talking about a human organism if we're talking about that part that is derived from human DNA alone. Organisms and superorganisms are defined by boundaries, and the persistence of a human being through space and time requires constant boundary repair. And this boundary repair occurs through immune responses. And as boundaries repair, novel processes emerge. What transplantation does for us is facilitate the understanding of health by coinciding structural and functional boundaries. And by doing so, health and personal identity emerge from the mode of arrangement of the superorganism's parts. And so we do have cells in our body, such as macrophages, which are very amoeboid in their characteristics. And so it is possible that these macrophages actually derived from other organisms, somehow managed to violate the structural boundaries of the human organism and integrate into the human organism, but at the same time preserve the characteristics of their amoeboid ancestors. Same thing goes with mitochondria. Many people believe that the mitochondria inside our cells actually derived from non-human sources. So even the human organism is constantly being redefined. So to summarize, overall, the human organism and microorganisms together constitute the human superorganism. Both organism and superorganism are contained by biological boundaries. These boundaries are maintained and repaired by the immune system. Emergent disease and health processes arise when these boundaries are disrupted and restored. So the immune system, unlike other organ systems, belongs to the superorganism. It does not belong to the organism. The role of the immune system is to keep the superorganism together. And this is why it interferes with transplant success. So the cardiovascular system belongs to the human organism. The respiratory system belongs to the human organism. The digestive system belongs to the human organism. the immune system belongs to the human superorganism. Unfortunately, our current transplant therapies direct towards the organism, and that's the source of all the problems. And so transplant success may improve if therapies are directed instead to the superorganism. So I'll stop there. Thank you, and I'll be glad to entertain questions.